Hello, Bio 100. This will be our lecture on the first half of Chapter 10, which covers basic patterns of inheritance or Mendelian inheritance. So we'll first begin with a brief description of who Mendel was. And so Gregor Mendel was an Austrian monk, and his original aspiration was to become a teacher. Interestingly, he was not able to pass the oral exams to become a teacher, and so he eventually became a monk, but he retained this interest in science and actually in math as well, apparently. And at the monastery, he was responsible for growing the garden. And in the garden, he not only grew vegetables, but he also did these experiments as he was producing food for the monastery. The organism that he worked with most frequently was the pea plant. And the pea plant was a good choice for him because it had very simple characteristics that were easily distinguishable. If we look at the table found at the bottom of our slide, we can see that the traits he looked at were things like tall, short, inflated, constricted, round, wrinkled, and so forth. These were traits that were easy to identify, and therefore they made it convenient for him to work with pea plants to do these genetic crosses. So when he did these experiments, he got these results that were confusing to him based on the prevailing view of the genetics that was present at that particular time. And we'll take a look at what these are in just a moment. What we do notice is that Mendel is significant, and we're talking about him today, because he formulated this idea of the so-called particulate theory of inheritance. Now, Mendel had no idea what DNA was. He had no idea what genes were. Those things would be discovered and named much later. But he came up with this idea that there was some sort of particle that was present in an organism that was passed from parent to offspring, and that particle contained something. We now know that those are genes that code for the traits of that organism. And he determined that these genes interact with one another in interesting ways. So basically what he noticed was the following. If we look down at the lower right-hand corner of our screen, we'll see that we'll see a representation of the cross, one of the crosses that he performed. So what he did was to take pea plants that had purple flowers and cross them with pea plants that had white flowers. This was the parental generation. In other words, the very first generation of this experiment. The offspring are referred to as the F1 generation, the offspring of that parental generation. And so he was surprised that in the F1 generation, he obtained all purple flowers in this particular experiment. That was surprising to him because at the time, people believed in what's referred to as the blending theory of inheritance. And so the blending theory of inheritance was nothing too profound. It simply stated that when organisms reproduce, the offspring are an intermediate. In other words, they're a blend between the two parents. This, of course, was not what he observed when he crossed a purple flower, as we see here, with a white flower and obtained all purple flowers. He then took two members of that F1 generation, in other words, both purple flowers, he crossed them together, and he obtained the F2 generation. In the F2 generation, he obtained 75% purple and 25% white. And so this was surprising to him as well, because obviously that is not consistent with the blending theory of inheritance. The blending theory of inheritance would, of course, predict all purple flowers in that F2 generation. So Mendel, this is a simple example, what we have on this screen, of what Mendel did. But if we go back up to the notes, what we notice is this. Mendel kept really large records. He had very large, what are referred to, as you all know, as N numbers. In other words, there were a large number of organisms that were present in these experiments. He was pretty good at math, as I mentioned, and had studied at the university with a physics professor and was very proficient at math. And his understanding of math helped him to identify these trends in the flowers of the pea plants and other traits of the pea plants that he was working with. So as we mentioned, Mendel's idea was the particulate theory of inheritance. Mendel called it the particulate theory of inheritance. The particles, as we now know, are chromosomes, and the chromosomes contain the genes. Mendel is referred to as the father of modern genetics for this particular reason. It's also interesting to note that Mendel was ahead of his time, and by that I mean that during his lifetime, no one recognized his accomplishments. No one recognized that he was correct in his formulation of this particulate theory of inheritance. This was not something that was known to science, and so no one thought that a monk could figure out something like this. And so he passed away, he died, and then a number of years later, this was rediscovered, so to speak, by a couple of scientists, and they realized, because of a paper that Mendel had written, that he had indeed been the first person to come up with this idea that genes code for traits, and those traits are passed on from parents to offspring and interact with one another in an interesting way. Now, as we begin our discussion here of this view of genetics, what we notice is this. As I mentioned previously, the particles in Mendel's particulate theory of inheritance are actually chromosomes. And so this, of course, is now the chromosomal theory of inheritance. The chromosomes are carriers of the genetic information, and so they are, of course, the carriers of genes. I think at this point in our semester, we're definitely familiar with genes and chromosomes, but if we look at this diagram from our text, what we notice is this. Let's just remind ourselves of the following. We have two pairs of homologous chromosomes that are shown in this diagram. On the left-hand side, the chromosomes are not duplicated. On the right-hand side, the chromosomes are duplicated. And so remember, don't forget, always remember that homologous chromosomes are not identical, but rather, as you see where I'm pointing here, homologous chromosomes contain alleles. Alleles are alternate forms of the same gene. So in this particular case, at this locus or location on the chromosome, we have a recessive allele, perhaps a recessive allele for hair color, and on the homologue, the other pair from the homologous pair, we have a dominant allele, perhaps a dominant allele for hair color. Perhaps this is black hair and this is red hair, but those are alleles. So homologous chromosomes are not identical, but they do contain alleles. Alleles are alternate forms of a gene, and those genes are located in identical spots along the chromosome. However, also remember this, that sister chromatids are identical. Sister chromatids are identical. Now that, of course, can be disrupted a little bit with crossing over, as we talked about in the previous chapter, but until crossing over occurs, sister chromatids are identical. Sister chromatids are identical, and of course, they would only be non-identical for a brief period in meiosis, as you know from chapter 9, and that would only be after meiosis 1 and then, of course, before meiosis 2. So remember, homologous chromosomes contain alleles, and sister chromatids are identical. Now, what we notice is this, that on this particular slide we have a very simple Punnett square that explains Mendel's initial observations. And so if we look up at the top, at this figure from our text, we see that we have a green pod from a pea plant and a yellow pod from a pea plant. The green pod is homozygous for green, the color green. In other words, it contains two alleles for green, and those alleles are dominant. In other words, they mask the yellow allele, which is therefore the recessive allele. We therefore represent the green allele with a capital G, and we represent the recessive allele, which codes for the color yellow in this case, with a lowercase g. Now, when the gametes are formed, each parent gets to supply one gamete to the one allele to the offspring. Each parent does supply one gamete, and each gamete contains just a single allele for a trait. And so in this particular case, the pea plant that has two dominant alleles can only supply a dominant allele. The pea plant that has two recessive alleles can only supply a recessive allele. And this means 100% of the offspring in the F1 generation are phenotypically green, but genotypically they have one dominant allele and one recessive allele. We refer to these individuals, these plants, as heterozygous, because they have one dominant allele and one recessive allele. Now, furthermore, what we notice is this, that the next thing Mendel did was to cross two plants from the F1 generation. In other words, they're both green phenotypically, and genotypically, they're both heterozygous. So, this means that when we create the gametes, each gamete can either receive a dominant allele or a recessive allele. And so there are two types of eggs, an egg that has a dominant allele that I'm pointing to here, and an egg that has a recessive allele. And there are two types of sperm, a sperm that has a dominant allele and a sperm that has a recessive allele. And so when the cross occurs, what we notice is this, a Punnett square, which is shown right here in the purple portion of our diagram, is a simple way of arranging the alleles. What we notice is that if this sperm meets this egg, then we have an individual that's phenotypically green, and genotypically we say that they are homozygous because they have two of the same alleles, and both those alleles are dominant, so we say they are homozygous dominant. These two individuals obtain a recessive allele from the egg and a dominant allele from the sperm. Well, I should say that one is the way I described, but this one obtains a dominant allele from the egg and recessive allele from the sperm. So the end result is they're both identical. In other words, this one is identical to this one, and they're both phenotypically the same as the homozygous dominant individual. They're both green. The phenotype is green, but genotypically they're both heterozygous. They're both heterozygous. In the lower right-hand corner, what we notice is that this individual gets a recessive allele from the egg and a recessive allele from the sperm, and so they are genotypically homozygous recessive, and phenotypically they are yellow. So the phenotype is what the organism looks like, and the genotype is the genes that code for that appearance. The genotype and the phenotype may differ. For instance, in this case, if you follow the pointer on the screen, this green individual, well, both of them are phenotypically green. Phenotype's the same, but the genotype's different. This one, homozygous dominant, this one, heterozygous. So what we see is the following, that in this particular case, the phenotypic ratio is 3 to 1, 3 green to 1 yellow, but the genotypic ratio is 1 to 1. In other words, 25% or 1 are homozygous dominant, 50% or 2 are heterozygous, and 25% or 1, again, the last one of the 4, are homozygous recessive. We can use the science that we just talked about on the previous slide to conduct something that's referred to as a test cross. So, as we mentioned previously, an individual that exhibits the dominant phenotype could have one of two genotypes. For instance, if you follow the pointer, this green individual is heterozygous, and this green individual is homozygous dominant. As we look at the plant, we cannot tell the difference between those two. The genes are invisible to us without some sort of genetic test. But without any kind of genetic test, we can determine the genotype of that individual by performing this thing referred to as a test cross. So if we cross this individual, which exhibits the dominant phenotype, with an individual that exhibits the recessive phenotype, then what we see is this. We get a roughly 50-50 ratio in the offspring between green and yellow. I would encourage you to do a Punnett square to make sure that you understand how that works. So if you think about it, then what has to happen is the following. The recessive individual can only give a recessive allele, but the individual that exhibits the dominant phenotype can provide 50% of the time the dominant allele and 50% of the time the recessive allele. If the green individual provides the dominant allele, the offspring's green. If the green individual provides the recessive allele, then the offspring is yellow. And so you get about a 50-50 ratio between green and yellow in the offspring. On the other hand, if the green individual is homozygous dominant, then that means that 100% of the time the offspring is going to exhibit the dominant phenotype. There's no possibility for that recessive phenotype to show up because the recessive phenotype will always be masked by that dominant allele. Here we see a heterozygous individual, and that heterozygous individual is what's always going to occur when we cross an individual that's homozygous dominant with an individual that's homozygous recessive. Mendel's laws, as we have discussed here, apply to humans as well. What we notice is the following, that there are a number of genetic traits that we refer to as disorders. These are conditions in which a mutation has occurred, and that mutation is passed on from parents to offspring. Some of these mutations are quite common, and a couple examples we'll talk about in the next slides are very common. Others are very rare, but they're always passed on according to the laws that Mendel set up. And so in this particular diagram, what we notice is that individuals that have the recessive allele, in this case, are affected individuals. In other words, this individual would have the disorder, and this individual that I'm pointing to here would be a carrier. And so a carrier is a term we use to describe an individual that has the mutated allele, which is recessive, so they don't show it. Phenotypically, they don't have the disease, but they can pass that gene on to their offspring. And if they reproduce with another individual that's heterozygous, for instance, if we follow the pointer, that's what's being shown here, then there is, as you know from the previous slide, there's a 1 in 4 chance, or a 25% chance, that the offspring will have the disorder. Let's look at some examples. Cystic fibrosis is the most common genetic disorder that occurs in the United States today. The most common, let me be clear, genetic disorder. So, of course, diseases like cancer, diabetes, move on down the line, heart disease, many of those have a genetic component. But these are diseases that are controlled very distinctly by either one or a couple of genes, and so you either have them or you don't based on the genes that you inherit. Cystic fibrosis is controlled by a single gene. And so if we look at the left-hand side of this diagram from our text, what we see is that we have cystic fibrosis is a mutation in a gene that codes for a chloride ion transporter. So what's supposed to happen is that chloride ions are transported across the epithelium of the lung, and water is transported with that. When the chloride ion transporter is mutated, chloride ions are not transported, and so water does not follow via osmosis across the membrane of the lung, and you end up with this thick mucus that builds up in the lungs. Cystic fibrosis is a serious disorder, and it has a significant impact on the expected lifespan of the individual. What we see in the United States is that approximately 1 in 24 people are carriers for cystic fibrosis. 1 out of every 24 people, in other words, would be expected to be heterozygous for cystic fibrosis. This means that the disease occurs when 2 individuals that are heterozygous produce an offspring, and every time they produce an offspring, there would be a 1 in 4 chance that offspring would have cystic fibrosis. The individual that's shown in part B of this diagram here is an individual that has cystic fibrosis. Cystic fibrosis results in a buildup of thick mucus in the lungs that has to be removed on a daily basis, and that's what that percussion vest is. It provides some stimulation to the outside of the body, which breaks that mucus loose, and then it's removed via the suction that is in that individual's mouth. What we notice is that sickle cell disease, formerly known as sickle cell anemia, is another example of an autosomal recessive disorder. The inheritance pattern is exactly the same as for cystic fibrosis, and even for, it follows Mendel's laws, as we've talked about over the last few moments exactly. It's simply a recessive gene. In this particular case, it's a mutation in the gene that codes for hemoglobin. And when hemoglobin is mutated, the hemoglobin molecules that are found inside your red blood cells, over 1 million of them, clump together. If you follow the pointer, this is a normal red blood cell. This is a red blood cell of an individual that has sickle cell disease. Sickle cell disease is extremely significant in terms of its effect on the human body. It does have a significant impact on the expected lifespan of the individual, even in developed countries with access to the best medical care in the world. And it is extremely serious if you don't have access to medical care. In fact, the lifespan is expected lifespan is extremely short, tragically, for individuals who do not have access in parts of the world where they do not have access to medical care. Now, I will mention this. There are several different mutations that do cause sickle cell disease. They're all single mutations. They all affect hemoglobin. But depending on the particular mutation you have, it may be more serious or a less serious version of the disease. Lastly, we have Huntington disease. And so Huntington disease is different than the previous two in that it's an autosomal dominant disorder. And so this means that an individual that has just one gene will have this particular disorder. This is a progressive disorder that leads in deterioration of neurons found in the brain. The patients appear normal until middle age, and then you have this sharp deterioration that occurs, and death follows soon afterwards. These individuals have typically reproduced by that time, and what we notice is that, frighteningly, there's a 50% chance that they would have passed this gene on to their offspring. Now, that's assuming that only one parent is a carrier for, not a carrier, I'm sorry, one parent has the gene for Huntington disease. It's very unlikely that two individuals would both have Huntington disease and would end up together producing offspring. Possible, but unlikely. So the normal scenario would be just one parent has it, and in that case, the offspring have a 50% chance, as you could convince yourself with a Punnett square, of obtaining that gene. And so it's a 50% chance because they only have to have one copy. As long as they have one copy of the gene, they have the disorder. Another thing to note is this: there are no carriers for an autosomal dominant disorder like Huntington disease. There simply are no carriers. There are carriers for autosomal recessive disorders, but not for autosomal dominant disorders.